about the chemical messengers, which are the neurotransmitters or hormones, and then the receptors associated with the peripheral nervous system, okay? All right, so when we talk about the neurotransmitters in the nervous system, there are usually two types we talk about, norepinephrine and acetylcholine. Consequently, we have two types of receptors, okay? We talked about the highly specific nature of neurotransmitters and their receptors. And so cholinergic receptors will only bind acetylcholine, and acetylcholine is one of the most abundant neurotransmitters in the body, whereas adrenergic receptors only bind epinephrine or norepinephrine, okay? It can bind both of those. More so norepinephrine, so it has a higher affinity for norepinephrine. And a lot of the epinephrine that's released can be turned into norepinephrine. Okay, so cholinergic receptors specifically bind acetylcholine, and adrenergic receptors will bind epinephrine or norepinephrine. And in brackets, it says hormone here, and make the connection that it will bind norepinephrine as a hormone. Now, what makes norepinephrine a hormone? Under what circumstances does norepinephrine become a hormone? When it's in the bloodstream, okay? So that's in brackets here, that it binds it as a neurotransmitter at a chemical synapse, but it also binds it in its hormone form when it's coming in the blood, okay? All right, now consequently, because of the specific binding, we have some neurons releasing one type of neurotransmitter and other neurons releasing another type of neurotransmitter, okay? Now, I want you to understand here that all preganglionic neurons release acetylcholine, all. Both the sympathetic preganglionic neurons and the parasympathetic preganglionic neurons. So if you ever are looking at a question that's asking you about what neurotransmitter is secreted from a preganglionic neuron, it's always acetylcholine. Now, when you look on the postganglionic neurons, depending on what division they're from, this is where the neurotransmitter will differ. So parasympathetic postganglionic neurons secrete acetylcholine, whereas most, and I'll talk about why this is most in a few slides down, most sympathetic postganglionic neurons secrete norepinephrine, okay? Hormones that are released from the adrenal medulla, again, epinephrine and norepinephrine, this is where they're entered into the bloodstream. This is where we have this adrenal medulla chromaffin cell acting like a postganglionic neuron. So you can consider both of these to be postganglionic neurons, okay? The chromaffin cell, we said, acts just like a sympathetic postganglionic neuron. Alrighty, so I'll say that again. All preganglionic neurons, irrespective of their division, secrete acetylcholine. All sympathetic postganglionic neurons, including our chromaffin cells, secrete norepinephrine, and then all postganglionic parasympathetic neurons secrete acetylcholine, okay? Okay, now this is a very important image. It helps to summarize the relationships here. So let's talk about this. We said that all preganglionic neurons, right, so this is specifically on the sympathetic side, and this is illustrating those three routes of communication that we talked about for the sympathetic system. The first one is from the CNS to the sympathetic chain to the effector tissue, from the CNS to the adrenal chromaffin cell, then to the effector tissue. Keep in mind, this is using the blood. And then the third route was from the CNS to another ganglion, not a part of the sympathetic chain. We call this a collateral ganglia, then to the effector tissue, okay? Now, because we said all preganglionic neurons release acetylcholine, irrespective of their division, hence all postganglionic neurons will have cholinergic receptors. Okay, think about the synaptic relationship here. Whatever is being released on the preganglionic side will cross that synaptic cleft, and it must be able to bind to whatever type of receptor is on the soma of that other neuron. And because all of the preganglionic neurons release acetylcholine, we always see cholinergic receptors on, on the postganglionic neuron or on the adrenal chromaffin cell, okay? I'll say that again. We have all three of our preganglionic neurons releasing acetylcholine. And so that acetylcholine specifically binds to a cholinergic receptor. So on the postganglionic side, we must have a cholinergic receptor in each of these areas, okay? Now, when we talk about the postganglionic neurons, in the sympathetic division, this second neuron secretes norepinephrine or epinephrine, okay? And so it's gonna bind to a different type of receptor on the target tissue. It's not gonna be a cholinergic receptor because cholinergic receptors cannot bind norepinephrine, okay? So on the effector tissue, in the sympathetic division, we must have adrenergic receptors. And adrenergic receptors can bind that norepinephrine or epinephrine. So please make that distinction. Let's look at the parasympathetic division. It's much more simple, right? Both of the neurons release acetylcholine. Both of the receptors are cholinergic, okay? But there's a trick here. So there are two types of cholinergic receptors. There are nicotinic receptors and there are muscarinic receptors, okay? Nicotinic receptors and muscarinic receptors. So whereas on the postganglionic neuron, we have a nicotinic receptor, and if we go back here, we'll see that these are all nicotinic receptors as well. On the effector tissue in the parasympathetic side, it's not a nicotinic receptor, although it's still cholinergic. It still binds acetylcholine, but it's a different type of cholinergic receptor. It's a muscarinic cholinergic receptor, okay? I really want to make this clear, so I'm going to go back to this slide and sort of explain this one more time. All preganglionic neurons release acetylcholine. They cross that synaptic cleft. They bind to a nicotinic cholinergic receptor. That second neuron will release a different neurotransmitter, norepinephrine or epinephrine. That'll cross the cleft again, and that will bind to an adrenergic receptor on the effector tissue, in the heart, in the lungs, in the blood vessels, okay, the glands, etc. In the parasympathetic division, both neurons secrete acetylcholine, okay, both neurons secrete acetylcholine. One binds on the postsynaptic side here on the second neuron to a nicotinic receptor, but then on the effector tissue, there is a muscarinic cholinergic receptor, okay? Let's talk about the difference between muscarinic and nicotinic. Now, a nicotinic receptor is an ionotropic receptor, all right? So it's bringing back ionotropic versus metabotropic, which we talked about in Unit 2, okay? So a nicotinic receptor is also a channel, right? And so when we have acetylcholine bind to this type of receptor, it opens up some sort of cation channel that causes a depolarization in the membrane, okay? Whereas a muscarinic receptor is a metabotropic receptor. It's going to be coupled to a G protein, and that is how it will bring about a cellular response. So remember that the nicotinic receptors are going to be faster. We talked about why ionotropic receptors are faster, because the receptors are also acting as a channel. Whereas muscarinic receptors, these are the receptors on that effector tissue, these are going to be a bit slower to bring about their response, but they will bring about a more amplified response because of the second messenger pathway that's sort of elicited here, okay? So a faster response and a slower response. Now let's jump back here and talk about why this is necessary, okay? Now we want a fast response in these areas, okay? So acetylcholine will bind to this nicotinic receptor that will open up a channel immediately, and that will depolarize this second neuron, okay? Or in the case of the chromaffin cell, that will help it to release that neurotransmitter. So this needs to be fast, right? Neuron to neuron, we need that second neuron releasing its neurotransmitter quickly. So this first receptor here is a much faster response. Whereas the receptors on the target tissue, including this muscarinic receptor, is a much slower response, okay? Adrenergic receptors and muscarinic receptors are metabotropic. They're going to be coupled to a G protein. They're going to have to elicit some sort of second messenger in order to bring about their response on that tissue. And so it's going to be much slower than it is on this second neuron. So that's why we have the different type of receptors in those different areas, okay? Fast versus slow. Okay, so let's talk about some location of receptors here. This is kind of what I just went over, but this is much more organized, a much more organized way of talking about it. So in the autonomic postganglionic neurons, there's always a nicotinic receptor because all preganglionic neurons always release acetylcholine, okay? Whereas on the effector tissue in the parasympathetic system, it's a muscarinic cholinergic receptor. On skeletal muscle, it's going to be a nicotinic cholinergic receptor. We'll talk about skeletal muscle later on. And then skeletal muscle also has a few nicotinic receptors here, cholinergic receptors on sweat glands. Now, the trouble comes up with sweat glands because there are different types of sweat glands. Okay, so sweat glands are part of the sympathetic response, right? Think about being nervous, palms are sweating, axilla is sweating, and things like that. And so these are unique in terms of the location of cholinergic receptors. Usually, we have sympathetic influence on our other glands, okay? So sweat glands are sort of an exception here. Okay, now we're gonna speak about the different types of adrenergic receptors. So there are more than one. There are several types of adrenergic receptors. There are two main classes, alpha and beta, and then underneath alpha and beta, there's some subclasses as well. Now, before we get into this, what I need you to know about the receptors is their varying affinities, okay? How do they differ in terms of the neurotransmitter that they bind? You also need to know their location, tissue locations, as well as their second messenger, okay? And we'll talk about all of that. But just sort of keeping in context what you need to know. So under alpha, we have alpha 1, alpha 2, and then under beta, we have beta 1, beta 2, and beta 3. Now, these are found in various locations of the body. They're not all in all of our tissues. We have alpha 1 on blood vessels, beta 2 in the lungs, beta 1 in the heart. So they're found in unique locations, and they bring about a unique response, okay? All right, let's speak about alpha receptors. They're usually excitatory, and when I say excitatory here, I mean their effect on the tissues. They usually are excitatory on their tissue, in their tissue effect. They're usually located in the sympathetic effector organs, so the heart, the lungs, blood vessels. These are the most common or the most abundant receptors. And this is kind of because the sympathetic response is more amplified, right? More widespread, more diffuse. They have a greater affinity for norepinephrine than epinephrine. Okay, so specifically, alpha receptors usually have a greater affinity for norepinephrine than epinephrine. And all that means is that they have more of an affiliation to norepinephrine, and they bind it much more easily. Let's look at beta receptors generally. So they all activate a different second messenger than alpha 1. Okay, and this is gonna get a little bit confusing here. I'm gonna try to keep it as straight as possible. They all use cyclic AMP as their second messenger. And then they have affinities for norepinephrine and epinephrine that vary. So they have varying affinities here. Some prefer epinephrine, some prefer norepinephrine. All right, let's talk about beta 1. So beta 1 is found on the heart and the kidneys. It's usually excitatory. When I say excitatory, it's usually going to speed up the heart rate, speed up the kidney function, okay? And it has an equal affinity for norepinephrine and epinephrine, meaning that it doesn't prefer either of the two. It binds them with equal affinity, okay? Beta 2 receptors are found on some blood vessels, usually on smooth muscles of the lungs is where we find beta 2, and it's usually inhibitory to the tissue. When I say inhibitory here, I mean that it's going to decrease the tissue function. What that looks like in the lungs is dilation of the bronchioles. And that actually helps to bring in more oxygen. We'll talk about what that looks like. But inhibitory on its tissue level, or at the tissue level, means that it's going to vasodilate that bronchiole. It's going to help that smooth muscle relax as opposed to contract. Okay? Contraction would be an excitatory response on the tissue level. Relaxation would be an inhibitory response on the tissue level. It has a greater affinity for epinephrine. So beta 2 sort of sticks out in this way. It prefers epinephrine over norepinephrine, and that has to do with some of its locations on blood vessels and in the lungs. Beta 3, however, is usually excitatory again, found in adipose tissue exclusively, and it has equal affinities just like beta 1, okay? So To summarize, alpha receptors have a greater affinity for norepinephrine than epinephrine. They are usually excitatory. Beta receptors, beta 1 and beta 3, have equal affinities for epinephrine and norepinephrine. Beta 1 is found in the heart and kidneys. Beta 3 is found in the adipose. And then beta 2, which is sort of the unicorn here, is usually inhibitory, right? That's one thing that makes it stick out, but also it has a greater affinity for epinephrine, okay? So it's very different from the other two beta receptors. Any questions on that? Any questions on the receptors? Yeah, yeah. There are some small exceptions in some areas, but for your purposes, it's excitatory. Okay. Also, in this context of inhibitory and excitatory, make sure that you're thinking about the tissue level. Okay. We're going to talk about what the effect they have on their second messenger, and it may differ from the effect it has on the tissues. Okay. So in the context of excitatory and inhibitory here, we're speaking about the tissues, the tissue level.